Alright, so we have glycolysis. And so what glycolysis is, again, is the splitting of sugar. So here I have sugar bowl. And so is it if I'm have my sugar bowl and my sugar, splitting of sugars, am I pounding it with a hammer? Not exactly. So we're going to be talking about what's actually going on in our body to break down all of these sugar molecules. Alright. Just kidding. Alright. So, first of all, the overall process, what is happening? Overall process, here we go. There we go. So glycolysis, again, in the cytoplasm, we're going to make some ATP. It's going to be made into this acetyl-CoA, which is kind of intermediate step, and going in the Krebs cycle, making our CO2 what we're breathing out. And that goes into our electron transport chain. Oxygen has to be used, produces water and ATP. It also makes an ATP in the Krebs cycle. But again, we're just going to talk about the Krebs cycle. Let me grab my little pointer here. So we're going to be talking, about glycolysis. So again, occurring there, matrix, the Krebs cycle, and across the cristae or the inner membrane for the electron transport chain. So glycolysis, really we're just going to start with a summary. Taking place in the cytoplasm. How many times do I have to say this? It takes place in the cytoplasm. Anaerobic. So what that means is it doesn't need oxygen. And means not. Aero means air or oxygen, not air. Doesn't use oxygen at all. It requires an input of 2 ATP. So in order to gain some ATP, we need to put some in. What's happening is glucose is split. Glucose, remember, it's C6H12O6. So 6 carbons. We're going to pay attention to those carbons. So glucose is split into 2 molecules. Pyruvate has 3 carbons. So if we go, we're going to have, I just put you back on me real quick. So if I have 6, here's my 6 carbons, we're going to be splitting into 2 pyruvates with each 3 carbons. And so 3 carbon pyruvates. Let's go back over here, or pyruvic acids. And our next one, so, and that's what this is showing right here, 6 carbon to 3. It also produces 2 NADHs. If you remember, those are energy carriers. We're going to want those later in the electron transport chain. And produces 4 ATP. So we had to put in some, but we gained even more. Pyruvate is oxidized. So oxidized is loss. So it's lost electrons to acetyl-CoA, and CO2 is removed. And so we're losing some. Here's our ATP that we gained. We gained an electron, so we reduced here and made it into pyruvate. And we'll talk about this. If it's anaerobic, it goes into fermentation. If it's aerobic, it's going to go into the Krebs cycle. So, just to recap, how many ATPs did glycolysis use? Right, 2. That was the input. How many does it produce? That's right, 4. So, big math here, guys. Net gain. So what do you have to do? 4 minus 2 gives you 2. Excellent. Great job. And here's just a quick diagram of everything that's happening. So we put in 2 ATP. Those turn into ADP. Remember, ADP is the adenosine diphosphate, 2 phosphates. We added that with glucose, broke it down into 2 pyruvates. If oxygen is not available, we're going to go into fermentation, which is the next thing we're going to talk about. If not, we're going to go into the Krebs cycle. And so here's where we made our ATP. We're going to talk about all this stuff in our next slides.